Hello, biology. Purpose of this video is to give those students who missed any of the 12.4 notes an opportunity to finish them up. Also to give those students who wrote their notes by hand an opportunity to get caught up, assuming I went too fast. Okay, this section is on mutations. It's 12-4 in the book. Remember, we always do our reading notes, our class notes in Cornell note style, which means questions on the left, answers on the right. We are starting these at number 25. These notes finish at number 40, which means you will need 11 rows. 11 rows and 2 columns. Okay, let's get started. All right, mutations. Any change in the DNA base sequence or any change in the order of nitrogen bases is considered to be a mutation. Typically, mutations take place when DNA replication has taken place. Remember, DNA polymerase comes in to add complementary nitrogen bases to the existing chain. And every once in a while, a mistake gets made. Now, remember, DNA polymerase also proofreads, but sometimes, obviously, mistakes are still made. Okay, we're going to talk about a couple types of mutations today. We're going to talk about gene mutations, and we're going to talk about chromosome mutations. Gene mutations are mutations that produce changes in a single gene. Makes sense. Okay, or at a single location on the chromosome. Because remember, genes are segments of DNA. All right, other type of mutation is a chromosomal mutation. And these are mutations that produce changes in whole chromosomes. We're going to abbreviate chromosome as capital CHR. Remember, I might be going a little bit too fast in this video, but that's okay because you can always pause the video. If you are not listening to the sound, the way that you will know I'm about ready to go to the next slide is I will point at the camera. Also, remember, we are starting at number 25. We are finishing at number 40. Do not start your numbering at number 1, or else I will take off points from your notes. I'm looking for 25 through 40 today, and that's it. All right, here we go. Moving ahead. All right, so point mutations. These are types of gene mutations that only involve one or a few nucleotides. Very specific and limited locations. There are 3 types. There's a substitution mutation, an insertion, which means adding, and a deletion, which of course means removing. Substitution does not add or remove, it just simply changes from one to the other. So an adenine could get changed to a cytosine. A thymine could get changed to a guanine. Okay, let's talk about what a substitution is. All right, so this is the DNA, so it usually affects no more than 1 amino acid. Okay, that's important. So this is the DNA code, all right? This is where the mutation takes place. So obviously, if it's a T now, it used to be an A, a G, or a C. But now it's a T, which means that when transcription takes place and messenger RNA is made in the nucleus, that is going to change our codon from C, whatever it was, U, to CAU, all right? And remember, the amino acids that get brought to the ribosome to make proteins depend on this codon for messenger RNA. They depend on the anticodon for transfer RNA, but of course that is complementary to this. So histidine, all right? Now, there is a possibility that this mutation, that CAU, maybe it used to be CUU, or maybe it used to be CCU or CGU, whatever the case may be. It's possible that that mutation, or pardon me, that original codon still coded for histidine. So it's possible, because remember, there are 64 different possible combinations of the letters, the nucleotide bases, and there are only 20 amino acids, which means that each amino acid is going to have at least 2 different codons. So substitution mutations are the least harmful types of mutations, because it is possible that the amino acid does not get changed. And even if the amino acid does get changed, it's possible that the protein will still be functional. Okay, moving ahead. Why are insertions and deletions more dramatic? Dramatic meaning more negative, more things happen. I guess that's what dramatic means. Okay, so the addition or deletion of a nucleotide causes a shift in the grouping of codons, and these are called frame shift mutations. Remember, each codon is a segment of 3 nucleotides. Excuse me. If you have an insertion that's going to add a nucleotide in there, it's going to shift every single nucleotide 1 over, which could very easily change every codon after the insertion. And if every codon is changed after the insertion, it's possible that every amino acid will get changed. And most likely that would make the protein non-functional, which could lead to some serious problems. Okay. A deletion, same thing. You've got your reading frame, your 3 codons that are going to be read at the ribosome, and all of a sudden one leaves, so everything shifts to the left. Same thing. It's possible that every single codon after that deletion is going to code for a different amino acid, again, making the protein non-functional, more than likely. Excuse me, sorry about the yawning. I did not sleep well last night. Okay, more information about that. They may change every amino acid that follows the mutation. They can change proteins so much that they will not function. And if it's an important protein, so remember, cystic fibrosis that we have, you have learned about in the movie. Cystic fibrosis, that protein allows salt to leave the cell, okay, lung cells. And if that protein is not functional, all of a sudden the salt can't leave the cell, which means that it builds up in the lung cells, which means it creates a mucus, which eventually in cystic fibrosis, more often than not, leads to death. So if it causes that protein to be non-functional, it's an important protein, then it could, again, my apologies, dramatically affect the organism. All right, moving ahead. All right, you guys, please use this time to practice with your notes. Don't fast forward until number 33. Okay, use this as an opportunity to practice with your notes. What are the 2 types of mutations? If you can't remember, take a look back at number 26 and 27. The 2 categories of mutations. You bet. They're gene mutations and they're chromosome mutations. What just happened? I have no idea what just happened. That was very odd. That's better. All right, point mutations involve changes in one or a few whats. And if you can't remember, take a look at number 28. You got it. One or a few nucleotides, which means that they are at very specific places in the DNA. All right, what are the 3 types of point mutations? By the way, I didn't mention this, but a point mutation literally is that. You can point and go, oh, it's right there. That A changed to a G. It's right there. specific locations. 3 types of point mutations. Take a look at number 29. All right, so you got the one that doesn't necessarily impact the protein, kind of depends, substitution, and then the 2 types that are frame shift mutations that likely will affect the functionality of the protein are insertions and deletions. Substitutions, insertions, deletions. Insertions and deletions are more negative because they are changing the reading frame. Every single codon after that mutation likely will be changed, and that likely will lead to a difference in amino acids, which likely will make the protein not functional, and that's why they're more dramatic. Okay, insertion deletions. All right, an extra base is inserted into a base sequence. So again, the reading frame shifts. So this is the original DNA. It used to be TAC. Now it's TATC. So this T was inserted into this DNA sequence. So now you notice instead of methionine, arginine, threonine, and leucine, it's now isoleucine, alanine, tyrosine. The only one that is the same is leucine. Okay, which means, and remember, proteins are, tens, hundreds, thousands of amino acids long. So it's very possible that this protein will now be non-functional. Uh-oh, my bad. And remember, when we say the reading frame, we're talking about the codon. Because remember, the codon, the 3-letter code, all right, which used to be AUG, now it's AUA. That 3-letter code is what's read at the ribosome, and that determines what anticodon in transfer RNA is going to come and what amino acid is being brought with it. All right, moving ahead. All right, deletion, same idea. I like this one because it actually gives you some words. The fat cat ate the rat. Now we delete this H, okay, which of course would be an A, T, C, or G in DNA. Now all of a sudden it's Tef, Atka, Atta, Tet, Her, At. Okay, so you can see how every single 3-letter code after that mutation is going to be affected. And again, the idea is that it likely will change all of the amino acids after the deletion, which obviously will make the protein non-functional. All right, moving ahead. All right, chromosomal mutations. So point mutations, insertions, deletions, substitutions, gene mutations, those are all the same thing. Chromosomal mutations happen on a much larger scale. Changes in the number of chromosomes or the actual structure of the chromosome. Remember, a chromosome is just a massive, massive organized piece of DNA, okay? Okay, there are 4 types of chromosomal mutations. Deletions, where all or part of a chromosome are removed. A duplication, where a segment of the chromosome is duplicated. Inversion, to invert, means to turn upside down. So that's when the bottom portion of the chromosome is now on the top, or a part of the bottom is now on the top. And then, of course, part of the top would be on the bottom. And a translocation, I honestly cannot remember what a translocation is. Yeah, we'll have to get there. There are some pictures, some diagrams that go along with these descriptions. That'll make it easier for me to explain to you guys. All right, getting ready to move ahead. Okay, deletion. So these letters, they don't make a whole lot of sense to you guys now, but when we start talking about genetics, segments of these chromosomes code for genes. So just think of the A gene, the B gene, the C gene, D, E, and F. A deletion is loss of all or part of a chromosome. So all of a sudden, the B gene is gone, which means that the cells that end up with this new chromosome will not have the instructions for the proteins that the B gene or the B genes coded for. Okay, duplication. All right, so we got A, B, C, D, E, F. All of a sudden, there's two B genes, all right, which might not be a big deal because that just simply means that there are more of those proteins that those genes code for. Although I'm certain it can't be quite that simple. And remember, what happens, again, this is kind of premature because we haven't talked about mitosis or meiosis. All of the DNA gets replicated. DNA replication takes place before cells split, before cells reproduce. Okay, so all of these types of mutations are going to happen prior to meiosis, which is the creation of sex cells, or mitosis, which is the creation of body or somatic cells. Moving on. All right, and inversion. We've got A, B, C, D, E, F. Now all of a sudden we got A, E, D, C, B, F. So these two got flip-flopped. Okay, so the C is now in front of the B, and it's on this part of the chromosome. The D and the E got flip-flopped, and they are now on this part of the chromosome. So taking part of, so usually chromosomes are seen top to bottom. So this would be the top of the chromosome. This would be the bottom of the chromosome. So switching portions of the top to the bottom and the bottom to the top, and these genes get flipped. And depending on, I think it's called a promoter. Promoter is what says where RNA polymerase starts making messenger RNA. So depending on those signals, those chemical signals, those enzymes, this might be a problem for the cell if things are in different orders. I'm not really sure how smart enzymes are when it comes to that type of thing. Okay, this is the one I could not remember. Translocation. So we got A, B, C, D, E, F. Okay, so yeah. So there's another portion of the chromosome that's not being seen up here. That's going to be GHI, JKL. So now all of a sudden, ABC, this portion is on the different chromosome. This portion that's normally here is on a different chromosome. Again, moving things to where they shouldn't be. And again, that may or may not, probably will, make creating those proteins more difficult. All right, moving ahead. All right, do all mutations affect gene expression? No, it depends. Substitution mutations, especially, might not even change the amino acid. That would be just one amino acid. So not all do, but some do, clearly. Are all mutations bad for the organism? No, depends. There are good mutations that may produce new proteins that can be useful. For example, evolution. The way that species change over typically thousands of generations, so really long periods of time we're talking about, are the accumulation of the mutations that happen in those organisms. Also, there's another source of genetic variation or another source of evolution that we'll talk about in a couple of weeks, and that is called crossing over. It's when the chromosomes, this is meiosis, when sex cells are being created, but when the chromosomes duplicate and they line up, the inner chromosomes sometimes cross over, and then the top part of this chromosome is now on the top part of this chromosome, and the bottom part of this chromosome is now on the bottom part of this chromosome. So they swap information. And sometimes that leads to new proteins, new genes. Okay, vocab word poly means many. Ploid, you wouldn't know this yet because we really haven't started talking about chromosomes yet. Diploid, haploid refers to chromosomes. So polyploidy means extra or more or many sets of chromosomes. And when we get to genetics, I'll teach you guys about the importance of polyploidy. Usually polyploidy organisms are a bit more rugged. They can handle changes in the environment more and still survive, usually. And my bad, I thought we were finishing at number 43. For those of you that are like, what do I do, Paulus? All you have to do is hit your tab when you are in the last box of your table, and it'll add a new row, it'll add a new row, and it'll add a new row. My apologies. I looked at the wrong box on my handout. Moving ahead. All right, during blank, a new base is added to a base sequence, where during a blank, a base is removed. If you can't remember, take a look back at 33 and 34. Yeah, when a base is added, it is an insertion. When a base is removed, it is a deletion. What are the 4 types of chromosomal mutations? Take a look back at number 36. Pardon me again. Yeah, so we got deletions, duplications, inversions, translocations. All right, a little matching. So you're going to want to take a look at number 33, 34, 35, 37, 8, 39, and 40 to do these. Okay, let's see if we can do the easy ones first. To invert means upside down. Let's see, that's going to be, well, deletion is going to be C. Loss of a portion or all of chromosome. Inversion is reverse, which is the same as upside down, so that's B. So 3 is B. Extra copies of chromosome parts, that's a duplication, so 2 is D. And that would mean that a translocation is A. Part of a chromosome breaks off and reattaches. Are all mutations bad for a species? Explain. If you can't remember, take a look at number 42. Of course, the answer is no. It really depends. It depends on whether or not those genes, those segments of DNA, are intact enough for proteins to still be made for the organism. Plus, we now know that mutations, along with crossing over, which we'll talk about in a couple weeks, lead to change in species, evolution, new characteristics that give those organisms an advantage. So no, they do not all mean bad things. All right, moving on. All right, a mutation in which all or part of a chromosome is lost. What do we call that? You got it. That is a deletion. A mutation that affects every amino acid following an insertion or deletion. What do we call that? Or changes the codon and how that codon is read at the ribosome. Those are referred to as frame shift mutations. Mutation in which a segment of a chromosome is replicated. To replicate means duplicate, therefore it's a duplication. The type of point mutation that usually affects only a single amino acid. This is the one that probably will not lead to a change in the protein. Yep, substitution. Last one, when two different chromosomes exchange some of their material, the mutation is called, the clue is two different chromosomes exchange material. Translocation. All right, ladies and gentlemen, that is it for the notes. I will see you in class in the Zoom.